Good afternoon, everyone. My name is Ben Forrest, and on behalf of Global Genes and the Orphan Disease Center, it's my distinct pleasure to welcome you all to this session titled Waste Not, Want Not, Making the Most of Small Patient Population Data. Thank you so much for taking the time to join us today. Before we get everything kicked off, I do want to review just a few brief housekeeping items. So the chat box to the left of the video allows you to interact with each other and the program staff. And we hope that you use that opportunity to make some new connections. You know, in these virtual formats, it's hard to do that. So use the chat box, make the best of that. Note that links and resources are not able to be shared in that chat. So if you want to upload any shared resources for the community, share those on the social wall. The question box is also located on the left of your screen. We encourage you to use the questions box to share questions with the presenters. This is your time to find answers and insights to help you advance your work as a partner in research. And know that the speakers will not be able to see any questions entered in the chat area. Rather, they'll be in the official questions section. So if we're not able to answer or address all of those questions, we'll do our best to follow up with you afterwards one-on-one. To the right of your video, you're going to see captions and translations. So to utilize that, expand that session and then select the language that you'd like to use. So big thanks to Horizon and Greenwich Biosciences for their support for those tools. And if you have any questions about the technology or the program, go ahead and look at the request support button in each session or the live support chat in the bottom right corner of all non-session pages. Finally, we encourage you all to participate as active listeners and take advantage of an opportunity to hear from leaders and experts here. And we've got 3 awesome ones, and ask questions of them. We want to ensure that you have created a relevant and impactful program. And we appreciate you sharing your time with us. Feel free to play as big a role in this as you would like. So I'm now pleased to introduce our first presenter, Esther. Go ahead and take her from here.
All right. Well, hello, and thank you for inviting me to speak here today. You hopefully are already seeing my slide. I have one slide only for this presentation, and this is to spell out the name of the disease we're most interested in, my name and contact information. So my name is Esther Hars, and I'm the president and CEO of the Schwachman-Diamond Syndrome Alliance, which I founded just a few weeks before the pandemic broke loose last year. I am a molecular biologist, and I did my PhD on cancer, and it's actually acute myeloid leukemia to be exact. And you may be wondering why I include that detail, but there's a specific reason for it. So years later, my second daughter was born. You can see her on the screen. And she was born with a rare disease called Schwachman-Diamond syndrome. We went through a year-long diagnostic odyssey, which I've now realized is actually relatively fast. We went through 3 major hospitals in the Boston area, probably more than 20 specialists and countless tests, and finally arrived at the diagnosis. And so this is relevant for my background because while Schwachman-Diamond syndrome, or SDS for short, affects many parts of the body, the most devastating feature is high risk of leukemia. And you guessed it, it's acute myeloid leukemia. And with devastating, what I mean is that in patients with SDS who develop this complication, and that's around 1 in 3 patients by age 30, it's pretty much untreatable. So that is really the driving force behind all we do to prevent this from happening. So after she was diagnosed, I went through a period of grief, as probably many of the other rare disease parents can relate to. And then once her day-to-day care needs improved, we started to have a lot of late-night conversations with my husband. What are we going to do next? in one option would be just lay back and enjoy the time we have, and hope that somebody else will discover a cure. Another option would be to donate all our money to an established research lab, hoping that they will find a cure. And the third option would be to be more strategic and really look what we can actually do strategically to enable more research and move research forward faster. And so, as I mentioned, I'm a molecular biologist who worked on this similar or related complications. My husband is also a scientist. We are actually biotech entrepreneurs. And I actually happen to like people and working with people too. So we decided to go with the third option of really engaging and being strategic in filling gaps and enabling more research. So one of the needs, one of the first starting points I explored was, well, are all the research tools available that are needed for therapy development? And as you probably all appreciate, mouse models are where it's at. Pretty much for most cures to happen, you need to have a mouse model for the disease. And so I read all the articles on SDS-related mouse models. I talked to all the researchers who were involved in developing them, and also talked to the researchers who are most likely to need a mouse model to advance their research further. And it turns out that there is no mouse model that is suitable for therapy development in our disease. And so we reached out to Jackson Laboratory to initiate a project to develop one. And we just kicked off this project very recently, and are very excited to be able to partner. And it's a true partnership with an institution where we bring the science and the connections and the collaborators into the mix, and they're bringing their expertise on developing the mouse model, and they're doing it for free for us. So that project is on the way. And in a similar train of thought, again, what can we bring to the table that's unique and valuable on the patient data and engagement side? And so what I don't want to do is to start just a random additional registry. Really, what our goal is to unite the different data sets that are already out there and add to it patient-reported data or data that are currently not being collected. So since the audience here is, or how should I say, let's cue back the session title. The session title is Waste Not, Want Not, right? So patient data may sound like something that's free because patients are not typically paid for them. But I would argue that that's actually the most limited and precious resource there is in the scientific endeavor. And that's because if patients are asked to submit data and then they don't see an impact, they don't see their data being used, no progress, they might just disengage and not engage in research in the future. So I would encourage you as the researchers to really engage with the patient community from the onset. Also have a plan for the longevity of the data beyond the particular project you're working on. That would include consenting patients in a way so that the data can be used for other purposes after the study, engaging with the patient community so that the data can be connected to other data sources and go further that way, and so on. And in an ideal world, really the whole registry strategy and framework should be managed by the patient community, because we are not vested in one particular project. Our ultimate goal is to find cures for our children and for our patients. And so we can have this long-term overarching goal of having this managing data and directing data. And so with the patient community managing the data, I really don't mean doing it at the kitchen table or an Excel sheet on a computer in constant contact, but really engaging with professionals who understand data governance, structure, strategies, collaboration, standards. So shout out to RareX. We really need to do it in a very responsible and thoughtful way to ensure that the patients get engaged and stay engaged until a cure is found. So, just checking. So overall, since the audience here is young investigators, I would say we want to help you succeed, and we define success as finding cures or improving the quality of life for our patients. So there is really, our goals can be really aligned and we can become true partners. And just, I haven't checked the time, but if I have one more minute, just wanted to throw something else out there. So you are young investigators, so you may be thinking about your longer term career goals or where your career should go. And just wanted to throw it out there that as the patient advocacy groups evolve and get more invested in research and directing research, there might be really opportunities for you to become partners with them and be a part of the patient groups, maybe as a science director or something like that. Because not every organization will have a scientist on board or somebody like Bina who can really learn and come up with strategies. And so if these organizations can hire young investigators or more experienced investigators to be really fully involved in directing research, that might really be a very rewarding career for you because you will be able to see real impact on patients' lives. And so I will leave you at that as the parting thoughts and hand the mic back to Ben.
Good afternoon, everyone. My name is Amy Waldman, and I am thrilled to be here today from my clinic space. It was a very busy clinic morning, so I apologize for all of the mess. This is not my office. But I'm here to talk about small patient data, and it's been a thrilling experience for me over the last 6 years to work with our Alexander disease and leukodystrophy population. And I wanted to just share some of the lessons. So starting with, sure I can advance there. There you go. So a lot of times we talk about where do you begin with patient data and collecting patient data. And we talk a lot about natural history studies, but I want to be sure that everyone understands that a natural history study is not the same thing as a patient registry. Both are critically important. Both have extreme use in our rare disease space, but they are a little bit different. So natural history studies are really considered the foundation of rare disease research, understanding change over time. So you collect information about all individuals who have a disease to provide an understanding of the clinical, the radiographic, genetic, and biochemical changes in the body. It's really to understand disease variables and disease outcomes. For example, as neurologists, what happens with seizures over time, or ambulation or gait, or those types of things. And it's really these natural history studies that are critical to clinical trial design. What is the patient outcome? On the other hand, a registry is a systematic collection of data, but it's not exactly the same thing. So you can have a registry or a patient list or a contact list, but usually it doesn't necessarily have all of those components I just mentioned as a natural history study. Still very critical, right? So we have a registry that has all the patients that contact us, whether they want clinical or research care. But it's not the same as our natural history study, where we're actually collecting specific data addressing certain questions. So a registry may collect information on numbers or type of patients, so how many patients have this leukodystrophy versus another one, or subtypes or phenotypes of a disorder. Newborn screening is a classic registry and following kids over time. And even there's registries for post-marketing of the drug looking for side effects. So what we did at CHOP is I started a natural history study for Alexander disease, but I actually broke our natural history study up into two parts. One, I call it our outcome measure study, and the purpose of that was to explore clinical trial outcomes. So gross motor function, fine motor function, speech and language. I'll show you in a few slides a little bit more about the study, where we also collected biomarkers and imaging. But I requested annual trips, if people could, to Philadelphia. Not everyone can come multiple times, but for those who are able to, we wanted them to come to Philadelphia so we can actually do a series of research tests. And this does require funding, but so that's where it gets a little bit more complicated. But we also have a natural history study. So my concern was maybe the people that are able to come to Philadelphia are the ones whose children are still able to walk or not wheelchair bound or live closer to Philadelphia. And so we might not get as many patients from the Midwest or internationally. I wanted to be able to get information on as many patients as possible. So we have what's probably more our registry, but our natural history study, which allows us to collect medical records. So it is a little bit more of, like I said, a natural history study, because we're collecting records, we're reviewing for certain milestones and change over time. And that, to me, reduces the bias of only seeing in person people that might have less severe disease. So this was how we set it up, just giving you an example of different ways that you can set up your own natural history studies. But this allows us to have this dual purpose, to collect records on patients who couldn't physically travel, but get a little bit more deeper dive into the ones that could. So as part of our Alexander disease study, we asked for spinal taps, and most of us are parents, and no one's going to necessarily sign their kid up for a spinal tap without communication as to why this is important. So one of the things that we were tasked to mention today is, what is the key to rare disease success? So first of all, you have to start somewhere, right? So we started with one patient, one advocacy group, and provided, luckily, excellent care and word of mouth spread, but you have to start somewhere. And what has been the most important to me has been the transparency with patients. So our story with Alexander disease, I found out that there was a promising drug being developed by a pharmaceutical company, and I was thrilled, and this is what everybody waits for and wishes for, but I was horrified at the same time, because I felt that we wouldn't be able to bring that drug to a clinical trial because we didn't have the patient data. And to me, that was unacceptable. The patients are there, right? But as Esther was explaining, we just didn't have the data on those patients. So to me, this was unacceptable. And we actually, within a month, did a retrospective study, and within 2 months had launched our prospective study to try to study this rare disease. And we just started small, and thanks to the Alexander disease patient community, who has been loyal and has come to visit CHOP, they've driven across the country, and to be part of this study, because of our transparency, I said, we need this data to get to the clinical trial. And we were fortunate that there was a drug in development in the preclinical stages. That, of course, helps quite a bit. But to Esther's point, even if there isn't a drug, the only way you're going to get to a drug is to have a systematic collection of data so that pharmaceutical companies know that there's a patient population that could be studied. So one of the things that I did too was I provided a rationale as to why I needed this information, right? And I want to talk in a few slides about the lumbar puncture. And I mean, I really was asking people to do something that was uncomfortable. And why did I need that data? And there was a very specific reason in that we were measuring the abnormal protein, and this was the only way to do it, right? We're not going to biopsy patients. You can't, we do it in the blood. It's not as sensitive as in the spinal fluid. And I'm going to show you why it's so important now in a few slides. There's also, the key to our success, we've had mutual benefit, right? So I hope our patients have benefited from coming to see, our team and disease experts, but we've also learned from them and been able to apply what we learned to other patients. So in the rare disease space, when some neurologists only see 1 or 2 of these patients over their careers, it's been tremendous to have, a few neurologists that are seeing a lot of these people throughout the country so that we can all share information and really knowledge and expertise. And I have to say, patient advocacy groups really do advance science. The NIH is wonderful, and I'm thrilled that we have some leukodystrophy support from the NIH, a rare disease grant that started about 2 years ago. But usually you have to start much smaller than that, right? Because you need to build up some data in order to show what your grant aims are going to be. And really, that's where patient advocacy groups are huge. Every big organization, whether it's Alex's Lemonade, which is a big pediatric cancer organization, or even like the MS Society or the cancer societies, those all started in grassroots and have been built to what they are today. And yes, of course, with larger number of patients, they can be built to much larger organizations. But our study has really been funded by philanthropy. And I have to really acknowledge the Alexander disease patient community and philanthropists that have really supported our study and really has moved the ball forward. And our clinical trial launched this month, which has been really exciting. But let me show you why this data is so important. And I show the Alexander disease families to say this is why we need this data. So let's say someone enters this clinical trial and we measure the protein in their brain, the GFAP is the abnormal protein, and we measure it at the entry into the trial, and then we measure it at follow-up. And we see that there's been an increase. And we say, wait a minute, the drug didn't work. The drug must have failed. The purpose of the drug is to lower this protein. It's gone up. The drug didn't work. But if we actually look at our natural history data from people who had put their child through an LP for the purposes of research, we see that the expectation was actually that their GFAP level would have gone much, much higher than it actually did when given the clinical trial drug, which really represents a decrease of 40% in the amount of this abnormal protein, and hopefully it continues to decrease further, and then we say the drug is a success. So with this type of transparency, explaining to families what we're doing with their data, explaining to families what questions we're trying to answer, has really been a tremendous reward, not only for the families, but for me as well, and we've really been able to engage our patients. But reliable data is key. And so we did a study where we looked at these longitudinal changes over time, and we noticed that the data looked all over the place. So sometimes it's going up, sometimes it's going down, sometimes it's going up, sometimes it's staying stable. But when we did a deeper dive, these data points were not collected at our institution and were not processed in exactly the same way. And so when you remove those data points, then you start to see some different trends in our data. Maybe there is a time of rise, a time of plateau, depending on when the patient was seen. And we have much more data now that we're analyzing. But it's really important that when you do these studies, that there is consistency, especially if you're collaborating with other sites. And then the last thing that I wanted to talk about is how do you choose what measures to study? We went broad. We picked gross and fine motor skills. We tests. We picked speech and language. I'll show you even more on the next page. So we did some performance measures, some clinical reported measures, like a clinical reported outcome a clinician reported outcome that I score, an observer reported outcome, and a patient reported outcome. So some different things so that I could score people, the family or caregiver could score them, and patients themselves could score themselves. So we put our patients through a very long list of scales. We did swallow assessments, we did self-report measures, we did these neurocognitive assessments. And this is my last slide. So my advice in terms of where do you start, right? We picked a broad number of scales, and we were fortunate that we had funding to do that. But when you're picking where do you start, you want to start with exploring existing scales. Even if the data wasn't collected in your disease, something that has been validated in your patient, in a patient population. So you at least know the metrics of the test are reliable. You, of course, consider the patient's age, ability, and change over time. That's, of course, key as well. Looking for whether you're going to have floor or ceiling effects. You know, is the test too hard? Is it too easy? And some of these tests that we picked, a lot of people couldn't do at all. And so the test was just too hard. And of course, we didn't know that when we started. Does the test need to be administered by a specialist? If you have funding, it's no problem. You have your physical therapist or somebody else administer it. If it does require some sort of training, that gets a little bit harder and may not be the place to start. And we didn't have everything going from day one. We picked some key things. We've added some things along the way. Yes, we can't analyze data that we don't have, but we did make some modifications along the way. And in the end, the most important thing when you're picking your patient data is to pick a meaningful clinical endpoint. So for example, there's the gross motor function measure and the 10-meter walk, right? So one is a measure of different skills within the gross motor domain. One is a measure of walking and endurance. We went to the FDA to say, here's the GMFM-88, and we're going to measure change over time in their gross motor skills. And the FDA says, we don't care. What is a 10% change or a 5% change? What does that number mean? We couldn't say to the FDA, it means you go from not walking to walking, right? So if we said a 20-point change, but then that would have been statistically hard to prove. So the FDA actually didn't like it. And we ended up on the 10-meter walk. So at the end of the day, you really have to say, does the test capture how a patient feels, functions, or survives? The FDA liked the 10-meter walk because they can say you improved their ambulation, you improved their endurance, but they didn't like the GMFM-88, so it was a lesson learned. And you really have to understand, how are you going to interpret change over time? What does a faster walk mean? What benefit is that to the patient, and trying to figure out how to interpret that. So I know I went very quickly, and I apologize. I want to make sure we have time for questions, but patients have been the key to our success. We didn't even get it right from the beginning. We added and stuff along the way, and keeping in mind the difference between the natural history studies and the registries. And I wish you all luck and well with your careers. And if there's anything that we can do to help support you, let us know. Thanks so much.
Hi, everyone. I just wanted to applaud Esther and Amy because I think they covered such important themes in the topic for today's panel. I am binashah, and I am the founder and CEO of Project 8P Foundation. My inspiration to leading this about 2.5 years ago is my wonderful daughter in this picture, who has chromosome 8P inversion, duplication, and deletion. Many have not heard of it, but we kind of fall in this category of neurodevelopmental chromosomal disorders. And as Esther mentioned, there are a variety of different things as a parent that, individually we come to terms with and have to grasp. And when I realized that my naivety of thinking about expert clinicians and researchers actively studying this disorder was non-existent, I realized I really needed to do something about this and switch careers and switch hats and try to learn about what science and medicine really is and how we can help better understand our disorder. And hopefully in the long term, identify therapies for us. So I wanted to share a little bit about our journey to starting Project 8P as a nonprofit to advocate for the community that's affected by chromosome 8P disorder. So again, it started with my beautiful little daughter, who is amazing, but is nonverbal, was a very late walker. It took her 6.5 years to have her first steps. She is progressing at what I call the inch stones, not milestones. And we decided to start a foundation. And our first assignment from one of the universities was to try to find 100 people affected by this disorder. So thankfully, to things like social media, there are some good things about it. We found more than 100 pretty quickly, all varying rearrangements within 8P. And I had to start learning what all of that meant and what it meant to really engage a community together behind why participating in research is so important. And a lot of that in the first 1.5 years was us just finding clinicians that would even just want to talk to us. They have never seen anybody with an 8P disorder or any even associated conditions, even not rare genetic disorders. And so we were fortunate enough with hundreds and hundreds of phone calls to academic universities saying, who can help us, who's interested, how can we help even fund and support some of the young investigators? And that's really where we ended up finding some really wonderful alliances, to say the least. And recently, unfortunately, when the pandemic hit, the lab shut down and we kind of had to go back to zero in terms of some of our cell lines. And so what we decided to do is launch our own IRB approved study for our retrospective and prospective natural history study, which also linked our biospecimens for our AP community. And based on the literature, there is an estimated 500,000 people affected. We have identified 350 people affected. I'm sure most of those are undiagnosed or misdiagnosed. We have achieved a little close to $1 million in revenue and funds raised, and we're all around the world, over 20 countries represented. And this was our first conference that we had in New York, where 63 families flew from all over the world, and there were 250 attendees. Within 6 months, we got everybody together. And this is when I said, raise your hand if you want to participate in research. And as you can see, everyone was cheering and raised their hands. And it was something that showed how important this was for our community. And we didn't really know, how important this could be and what the potential is. And then the next step was really connecting the dots. And so we found some papers. Some of them were very old. One of them here that I just grabbed a screenshot of was if 8P was a potential hub for neuropsychiatric disorders and the implications based on some genes for schizophrenia, autism, cancer, and so on and so forth. And so what does that really mean? There was data hidden in some old literature, tables that wasn't necessarily mineable electronically in exhibits, and then trying to find out if any researchers had any unpublished or negative results that they could share. One of the recent New York Times articles talked about whether scientists can really crack the code for disorders with genomic data. And one of the biggest highlights in that article was Moderna and how they kind of targeted the COVID-19 vaccine. They actually started working with the data set. So they treated it like a software problem without actually having the live virus in their lab. And again, I always thought about this as a business person coming into this, that are there successful in silico approaches to kind of look at the genes and the genomic data sets and even potentially identify candidates or matched pathways. And there's a lot that we don't know, and that humans, at the end of the day, it's just challenging, and the bandwidth is really hard because we're all, as patient groups, working with a finite set of resources, from funding to dedicated scientists and clinicians. And so we wanted to kind of demystify some of the myths about starting having a data platform. And myth is like, does it really cost millions of dollars? And that's what I was told. And so I just thought, not possible. It is possible. And another myth is, if you build it, they will come, but you have to build it with the right governance and compliance, and to build it so that patients are empowered so that they can benefit from this in real time, not 10 years from today or the unknown, to improve their own care and be able to share this data with their physicians. And what if we could build it so that clinicians and researchers like yourselves don't sit on outdated information? And so you may be working on a patient cell line 3 years later, thinking you have human phenotypic data to match your cellular phenotypic data, and then realize that that patient may not be living, or had developed an onset of 3 other comorbidities that really would have impacted your in vitro experiments. So, our wish list, this is a small snapshot of our wish list, is what could be possible if we joined forces. And, there are things like all the different sequencing data sets and the omics, and how clinics can exchange data easily on patients, and how can we compute and normalize and standardize unstructured data. There's a long list, and I have a limited amount of time today, but I'm happy to share this with you all and exchange the, and add to this. And so what I tried to do with our IRB approved study is launch the Chromosome 8p registry. And don't worry about the flow. This is really just about all the steps in terms of how you can optionally consent in or out of different components of the study. And in the center is really about answering a set of standardized survey questions that are patient reported, connecting it to some real world evidence through wearables, photos, videos, and including imaging like MRIs and EEG data, beyond just the clinical report, which for us usually says inconclusive. And then how do you really do this longitudinally? And then on the right, there is this option to consent and to provide your biospecimen. And then there's components to that of standardized protocols for all the way from initial collection and logistics, all the way to how do we reprogram into iPSC lines under the same standardized protocol so that we try to reduce variability from lab to lab. And then an additional whole genome sequencing, RNA-seq, and various omics that we could run. And so kind of making sure that we have all of that covered in the way that we designed the study. On the left, there is an additional feature to link to electronic health records. And each participant can decide to opt in, opt out, and what they wanted to share. And then again, furthermore to this left of the screen is, as we think about having multidisciplinary clinical care, how can those clinics report back what they've observed that may not be in the EMR back into the study? And then this big purple box at the bottom is really connecting all of these verticals. And so, we're really trying to make sure that this is accessible to approved clinician and researchers based on certain permissions, and to really make this as easy as possible. So with that in mind, with 2 other patient organizations, we also launched this commission on neurodevelopmental copy number variants, with a mission to think about tackling all of our resources and prioritizing treatment for those living with neurodevelopmental chromosomal disorders, and how can we really advance the field. And one of the biggest things that came about during this wonderful Zoom call period of last year is that we can really differentiate ourselves if this is patient-led and patient-centered, because there's so much information and engagement that can happen that's foundational to making sure that all of the grants that are being sought are most efficient and maximized, frankly. And how do we make this open access and scalable to other disorders? Because we are large copy number variants again, and there's quite an array of disorders within our rearrangements. So what we thought was, okay, it's lovely that we've designed a Chromosome 8p registry, but let's take it to another level, an interoperable data platform across disorders. And Esther mentioned RareX, and so we're working with the governance under RareX and designing a platform with Matrix, who you may have listened to Jason Colquitt in one of the other sessions earlier today. How do we do this as a platform approach, giving access to all of our stakeholders, first and foremost, patients and caregivers, and making sure they're contactable, and how can this help manage their own data? Clinicians and researchers and foundations, and I'm going to go through this a little quickly. The biorepository, which I mentioned earlier, and really about standardization and making decisions so that we differentiate into appropriate cell types based on the disorders, and all the different additional analyses that would need to be performed on that. And for us, most likely, those are 2D and 3D neuronal cells and organoids. And then running all of that raw data and making sure that raw data links back all the way to that patient-reported demographic survey to be able to make this really accessible to researchers in an informed way. And then what we're really trying to do is now, that's lovely to have a big, large data set, but to have that in some sort of web-based browser so that you can actually have analytics performed on this in addition to certain machine learning algorithms that can help make more informed decisions. And so with that, I will pass the mic. I'll drop the mic and pass the mic back to Ben.
All right. Thank you, Bina, for dropping the mic and passing it back. Very much appreciated. And thank you to the three of you for giving such excellent talks. I really highlighted the role of patients in research. Dr. Waldman, I'm so grateful for your distinction between registries and natural history studies. As someone who directs a free registry called CORDS, I'm like, I talk about that all the time myself, and I'm just so happy to hear other people out there saying the good word. And Bina, you're doing so many good, amazing things with Project AP. There's so much that everyone can learn from. So thank you all for sharing your insights. We do have some questions from the audience in a few minutes to answer them. So really quickly, I'd like to hear your thoughts on a question from Pamela Barraza Flores, which is, I'm a postdoc working in rare myopathies. I'm very interested in engaging with the community, but I'm not sure what's the best approach. So what's your best suggestion or tip for reaching out and engaging? I'll give each of you like 10 seconds. What's your thought, Esther?
Just reach out to, if there's a patient organization out there, reach out to them first, and they may be in a position to help or not. If not, maybe you can help them get there, and that's where I would start.
I agree. I would start with a patient advocacy group if there is one. If not, talk to a few of your own patients and see if you can spread word of mouth or bring those couple patients together, and then it will spread.
Yeah, I agree. And I think that there's always room for creativity. We didn't even know to work with like young investigators. And so when people have come to us, I'm like, oh, let's brainstorm together and think about how we can do this. Maybe there's just a one-off project, or I can get you 10 families and you can start learning more about this. So yeah, we're very much open door.
Awesome. Well, thank you again. And to the attendees, thank you. Before you leave for the next session, please take a few moments to respond to the polls that are being launched right now as we speak. And thank you again to our wonderful presenters. We appreciate all the feedback on this session that we want to ensure that you have the ability to provide the content and programming that suits your needs. You can share that feedback via events@globalgenes.org. Please let us know how everything went. And join us today at 3:30 Eastern Time, aka 1 minute from now, for office hours and informal networking. Those of us that'll be there will be there for as long as we can. And additionally, we encourage you to check out the agenda for the remainder of the week. There'll be office hours tomorrow as well. And there's a Cure Accelerator live pitch event at 4:30 Eastern Time today. So Make sure to take some time to explore that events page, visit the posters, build your itinerary for the rest of the week, and really just set yourself up for as good of a virtual conference as you possibly can. Then, last but not least, visit the Achievements and Badges tab to learn more about how you can win a pass to a future Global Genes program. How cool is that? So thank you so much to everybody for joining today. Just like that, the session is over. I wish you all the very best health and well-being, and take care.